Welcome to the deep dive. Today, we're going deep into something pretty fundamental, something your body's doing all the time without you even realizing it.
Right.
Your immune system.
Yeah.
And we're going to be using this awesome study guide that you sent over
Yes.
as our jumping off point to explore how our bodies put up these incredible defenses.
Exactly. It's really like a behind-the-scenes look at your body's security team in a way.
Yeah.
And the guide that you provided does a great job of breaking down the two main branches of that system.
Yeah.
We have that kind of rapid response team, the innate immune system.
Right.
And then we have the more specialized forces of the adaptive immune system that learn over time.
Right.
So using that framework, we can really dig deep into this topic and pull out the most important and relevant insights for everyone.
Okay, so let's start with that immediate protection then.
Okay.
The innate or non-specific immunity.
Right.
The guide starts by highlighting those surface barriers.
Yeah.
So we're talking about the skin, right? That's our first line of defense. But it's not just a passive wrapper.
Not at all.
It's really doing a lot.
The skin is a remarkable barrier. If you think about it, we have multiple layers of skin.
Yeah.
And those layers are tightly connected.
Right. So it really is a wall.
It's a wall, yeah, keeping those invaders out.
Yeah.
As long as that wall remains intact, it's a very, very effective first line of defense.
Okay. And then we have the mucous membranes.
Right.
I mean, sounds kind of gross.
Yeah, but it's so crucial.
Yes.
I mean, think of it as a sticky trap for anything that shouldn't be in there.
Right. Exactly.
The mucus itself is going to trap unwanted microbes.
Yeah.
And then you have cilia.
Yeah.
Those little tiny hair-like projections.
I've heard of those, yeah.
They're constantly working, in places like your respiratory tract, your nose, constantly sweeping that mucus away, taking all those unwanted things with it.
So it's just like this constant cleaning process that's happening?
It is a constant cleaning process, yeah.
That's pretty cool.
Yeah.
The study guide also goes into this whole other collection of barriers, too. We've got, tears washing our eyes, saliva in our mouth.
Right.
Even sebum, that oily stuff on our skin.
Yeah, it all has a purpose.
It seems like our body just has all these localized defenses that are just always on guard.
It's really a very comprehensive initial defense.
Yeah.
Even your tears, right? They contain enzymes that can actually break down bacterial cell walls.
Wow. I didn't realize that.
Yeah, and saliva has its own antimicrobial properties. And then that oily sebum that you mentioned, produced by your skin glands,
right.
It creates this slightly acidic environment that a lot of these microbes just don't like. They can't thrive there.
Right.
Plus, it forms a nice protective film.
Yeah.
Then, of course, you have the very acidic environment of the stomach,
right.
which most bacteria can't survive.
Yeah, that makes sense.
And the acidity in the vaginal tract also plays a really important role in preventing those harmful microbes from just growing out of control.
Okay, so that's a lot of barriers.
It is, yeah.
But there's this part in the guide that I found so interesting.
Okay.
It talks about Naegleria fowleri.
Ah, yes.
As something that can actually get past these surface barriers.
Right.
So what makes that pathogen so special, so unique?
Well, Naegleria fowleri,
yeah.
It's a single-celled organism that can actually enter the body through your nasal passages.
Oh, okay.
Often during recreational water activities.
Oh, interesting.
But what's really unusual is that it can travel along the olfactory nerve, the nerve responsible for your sense of smell, all the way up to the brain.
Oh, wow.
So it basically bypasses those initial surface defenses in a very specific way.
Right, right.
It just goes to show that even our really robust first line of defense isn't foolproof.
Yeah, there are always exceptions.
Right.
Well, that makes it even more important that we have those second-line internal defenses then.
Absolutely.
Which brings us to those phagocytes. The cells that literally gobble up and digest those invaders.
Yeah.
Like tiny little Pac-Man.
I love that analogy.
What are the key players there?
Well, the guide mentions macrophages and neutrophils. Macrophages, they can kind of be stationed in specific tissues, or they can be wandering around the body constantly on patrol.
Like guards, yeah.
Yeah, and then the neutrophils,
yeah,
they're typically the first ones to show up at the site of an infection.
Okay, so they're the first responders.
They are, yeah.
And the process of this phagocytosis.
Right.
It's a pretty detailed sequence.
It is.
First, there's chemotaxis.
Right.
So the phagocyte is basically chemically drawn to the trouble area.
Exactly. It follows a chemical trail, signals that are released either by the pathogens themselves or by damaged tissues.
Like a distress signal.
Exactly, like a distress signal.
And then what happens?
Well, next comes adherence.
Okay.
Where the phagocyte actually physically binds to the microbe.
So it got to get a hold of it.
Yeah, it's got to grab onto it.
Then it's ingestion, right?
Right. The pathogen is ingested, it's brought inside the phagocyte into a membrane-bound vesicle. It's like a little pouch.
Like it's swallowing it up.
Exactly.
Then we have phagolysosome formation.
Yes. This is where things get a little less pleasant for the microbe.
I bet.
The vesicle containing the microbe, it fuses with a lysosome, which is basically filled with powerful digestive enzymes that break down and neutralize the pathogen.
Yeah, it doesn't stand a chance at that point.
Right.
So then any waste product is just expelled from the phagocyte.
Exactly, through a process called exocytosis.
Okay, so that's the process of phagocytosis.
It is, yeah.
But getting those phagocytes to where they need to be, that sounds like a huge operation in itself.
It is quite a process.
The guide talks about leukocytosis, margination, diapetesis, and then more chemotaxis.
Right.
It's like this incredibly coordinated way of calling in reinforcements to the right spot.
It really is. So first, you have leukocytosis, which is basically an increase in the number of neutrophils in your bloodstream.
So that's like sending out more troops.
Exactly. And then you have margination, which is when those neutrophils slow down and adhere to the walls of the blood vessels near the infected area.
Okay.
And then the really amazing part is diapetesis. They actually squeeze through the gaps in the capillary walls to reach the infected tissue.
That's incredible.
It is, yeah.
And then, just like before, they follow those chemical signals.
That's right, chemotaxis again.
But, you know, the guide makes this point that not all pathogens are so easily defeated by these phagocytes.
Oh, absolutely not.
Some of them, they've figured out ways to resist, right?
Oh, yeah. Over time, they've developed some very clever strategies.
Like Mycobacterium tuberculosis, for example.
Oh, yes. Mycobacterium tuberculosis is a great example.
What does it do?
It can actually survive and even reproduce inside the phagocytes.
Yeah.
You see, it prevents the fusion of that vesicle, the phagosome, with the lysosome.
So it avoids those digestive enzymes.
Exactly. It hides out inside the phagocyte.
It's like its own little safe house.
Yeah, exactly.
That's amazing.
It is, yeah.
And then Streptococcus pneumonia has that slippery capsule.
Right.
Making it really hard for the phagocytes to even grab onto it.
Yeah, that capsule acts like a shield. It's really hard to get a grip on.
So it prevents that adherence.
Exact.
And it's interesting that N. fowleri, that brain-eating amoeba we talked about, it's mentioned again here.
Yes.
As sometimes not triggering that phagocytic response at all.
That's right.
So it's like it can evade even this second line of defense sometimes.
It can, yeah, in some cases.
It really does highlight that constant battle.
It really does, yeah.
This evolutionary arms race between our immune systems and these pathogens.
It's fascinating, isn't it?
Yeah, it really is.
This back and forth, this constant struggle.
Yeah, really cool.
Yeah.
Okay, so beyond those phagocytes, the innate system also has these natural killer cells, or NK cells.
Right, the NK cells.
Those sound like the body's internal security force.
That's a great way to think of them.
Dealing with any cells that have gone rogue.
Exactly.
So how do they work?
Unlike phagocytes, they don't engulf their targets.
Okay.
They identify abnormal cells, like cells infected with a virus or cancerous cells.
Right.
And they basically release chemicals that tell those cells to self-destruct.
Oh, wow. So it's like a targeted assassination.
It is, yeah.
Okay. Now, we can't talk about innate immunity without talking about inflammation.
Right.
We all know the classic signs.
Yeah, the redness, the heat, the swelling, the pain.
It's never fun.
No, it's not, but it's a really important process.
Right, so what's actually going on at the cellular level to cause all of that?
Well, it's all about containing the damage and starting the repair process.
Okay.
So when tissues are damaged or they encounter pathogens, they release all these chemical signals, and those signals trigger vasodilation. Yeah, I've heard of that.
Which is the widening of blood vessels in that area.
So more blood flow.
Yeah, more blood flow, which brings more immune cells and proteins to the site.
Right.
That's what causes the redness and the heat you feel.
Okay, that makes sense.
And those blood vessels also become more permeable.
Right.
So fluid can leak out into the surrounding tissues, which is the swelling or edema.
Okay.
That fluid helps to dilute any harmful substances.
Right.
And it brings in clotting factors and antibodies.
Wow, so it's kind of a multi-purpose fluid.
It is, yeah.
And then we see diapetesis again with those leukocytes moving out of those blood vessels.
Exactly. They're going right to the site of the problem.
Yeah.
The casticides come in and engulf the pathogens and the debris, and then finally the healing can begin.
Okay, so it's like a really coordinated process.
It is, yeah.
And the pain, that's just a reminder to protect the area.
Yeah, it's your body telling you to take it easy.
Right, exactly. Yeah. It's caused by those chemicals being released and the pressure from the swelling.
Okay, so the innate system also has these antimicrobial proteins, right?
Yes, it does.
Complement and interferons.
Right.
What is complement? The guide mentions there are about 20 of these proteins just circulating in our blood.
That's right. The complement system, it's a really potent part of the innate immune response. I mean, it can be activated in two main ways.
Okay.
There's the classical pathway, which is usually triggered by the adaptive immune system.
Right.
When antibodies bind to antigens.
Yep.
And then there's the alternative pathway, which is a more immediate response.
Okay.
Triggered directly by things on the surface of the microbes themselves.
Interesting.
Even those slippery capsules we talked about.
Right.
They can trigger this pathway.
So the innate system can recognize these features and just launch a defense.
It can. Yeah.
That's pretty amazing.
It is, yeah.
And then this complement system has a triple action, right?
Yes. It is.
Lysis, enhanced inflammation, and opsonization.
Right.
It can directly destroy microbes, enhance inflammation, and make those microbes easier to engulf.
Exactly. So lysis, it's the direct killing of microbes.
Okay.
By basically forming pores in their cell membranes, causing them to burst open.
So they just explode.
They do, yeah.
Wow.
And then complement proteins also act as inflammatory mediators, further boosting that inflammatory response.
Okay.
And then opsonization.
Yeah.
Think of it as putting a tag on a microbe.
Right, like a wanted poster.
Exactly, making it much easier for phagocytes to recognize it and gobble it up.
So it's like making those pathogens more appealing to the phagocytes.
Yeah, you could say that.
Okay, so that's complement.
Right.
And then we have interferons.
Yes, interferons.
Those are the messengers that help protect us against viruses.
Exactly.
How do they do that?
Well, they're released by cells that have been infected by viruses.
Okay.
And then they signal to neighboring cells.
Start producing your own antiviral proteins.
Oh, so it's like a warning system.
It is. Yes.
Helping to contain the spread of the virus.
Exactly.
Very cool.
Yeah.
Finally, within these innate defenses, we have fever.
Right.
That increase in body temperature. It always feels so miserable.
It does, but it can actually be beneficial.
How so?
Well, a moderate fever can actually speed up a lot of processes in your body, including immune responses.
Oh, okay.
So it can potentially make them more efficient.
Interesting.
And there's some evidence to suggest that it can actually inhibit the growth of certain bacteria and viruses that are sensitive to those higher temperatures.
So it's like turning up the heat.
Exactly.
Literally.
Yeah, literally.
Okay, so that was the innate immune system.
Right.
Or first and second lines of defense. Now let's move on to the adaptive immunity, right?
Yes, let's do that.
This sounds like the more tailored, long-lasting response.
It is, yeah.
Yes, the part that really learns over time.
It develops over time after you've been exposed to specific pathogens. And its hallmark is that ability to recognize and target specific invaders.
Right.
And then remember them for the future to provide that long-term protection.
Okay.
So within adaptive immunity, we have two main branches. Humoral immunity, which is all about B cells and antibodies.
Okay.
And then cell-mediated immunity, which involves those T cells that directly interact with infected or abnormal cells.
Okay, so before we get into those cells themselves,
the
study guide defines a few key terms.
Right.
Antigen, immunocompetency, and self-tolerance.
Yes, let's break those down.
Okay, so an antigen
Yes.
is basically anything that can trigger an immune response.
Right.
It's an antibody-generating substance.
It is, yeah.
So it's like the unique identifier of a foreign invader.
It is, yeah. It's like a little name tag that the immune system can recognize.
Right, exactly.
And react to.
Okay, so then what is immunocompetency?
Well, immunocompetency is the ability of your immune system to recognize those foreign substances.
Right.
And to actually mount a response against them.
So it's basically the ability to tell self from non-self.
It is, yeah.
And then self-tolerance.
Right.
This seems super important.
It is absolutely crucial.
It's the immune system's ability to recognize and not attack our own healthy tissues.
Exactly.
So it's like it knows not to attack itself.
It does, yeah.
And when that breaks down,
right,
that's when we get autoimmune diseases, right?
That's right. It's a delicate balance, that's for sure.
Yeah. It really is.
Yeah.
Okay, so now let's talk about those lymphocytes.
Okay.
The B and T cells,
Yes.
they mature in different locations in the body, right?
They do, yeah.
Where?
So B cells, which are responsible for producing antibodies, they mature in the bone marrow.
Okay, and the T cells?
T cells, which are key for cell-mediated immunity.
Right.
They mature in the thymus.
Okay, so they've got their own little training grounds.
They do, yeah.
And then we have these MHC proteins.
Ah, yes, MHC proteins.
Major histocompatibility complex proteins.
Right.
They're also known as antigen presenters.
Yeah.
It sounds like they're showing little bits of what's going on inside or outside of the cell to the T cells.
That's a really good way to think about it. They're like little display cases on the surface of your cells.
Yeah.
They bind to pieces of proteins
Okay.
from inside the cell for MHC class I and from pathogens that have been engulfed for MHC class II.
Right.
And then they show those pieces to the T cells.
Right.
So it's all about making sure that the right immune cells are activated against the right type of threat.
So it's like they're showing off a sample.
Exactly. They're presenting evidence.
The guide gives this helpful mnemonic. MHCI is for me. MHCCI is for you.
Right. A good way to remember it.
Yeah, can you explain that a little bit more?
Sure. So MHC class I proteins,
Yeah.
they're found on pretty much all nucleated cells in your body.
Okay.
That's the me part. And they present fragments of proteins that are made inside those cells. So if a cell is infected with a virus or it becomes cancerous, it starts making abnormal proteins.
Right.
And those abnormal proteins will be displayed on MHC class I's.
Okay.
And that basically tells the immune system, hey, something's wrong in here.
So it's like a flag.
It is. Yeah, it's a red flag.
Okay, so that's MHCI.
Right.
And then MHC2, that's the U part.
Yes. MHC class II proteins,
Okay.
they're found mainly on immune cells,
Right.
like macrophages,
Right.
dendritic cells,
Yeah.
and B cells.
Okay.
And those cells, they engulf foreign invaders.
Right.
They break them down,
Yeah.
and then they display those fragments on MHC class II.
Okay.
So it's like they're saying, hey, look what this foreign invader looks like.
Right, so it's showing it to other immune cells.
Exactly.
Okay, that makes a lot of sense.
Good.
So MHTI is like, look what's going on internally.
Right.
And MHCT is like, look what I found out here. Okay, now let's get into humoral immunity. And those B cells that produce antibodies,
Okay.
they recognize and bind to specific antigens using their B cell receptors, or BCRs.
Yes, that's the first step.
Right.
Each B cell has these unique BCRs on its surface that are specifically designed to bind to a particular antigen.
Okay.
So when a B cell encounters its matching antigen and the BCR binds to it, that triggers clonal selection.
So that B cell starts making copies of itself.
Exactly.
That are all specific to that antigen.
The activated B cell starts dividing, making a whole bunch of identical B cells, all with that same BCR that recognizes the initial antigen.
So it's like an army of clones.
It's
all ready to fight that specific threat.
Exactly.
And what happens to those clones?
Well, most of them become plasma cells.
Okay.
And plasma cells are basically antibody factories.
So they're pumping out antibodies.
Yes. They secrete huge amounts of antibodies into the body's fluids,
Okay.
the blood, the lymph, right?
And those antibodies circulate throughout the body, looking for their target antigen.
Right, like little guided missiles.
You could think of it that way, yeah.
But it's important to remember that the antibodies themselves, they don't directly kill the pathogen.
Not usually, though.
They're more like helpers.
They are, yeah.
They neutralize, mark, or clump those invaders
Right.
for other immune cells to take care of.
Exactly.
Okay, and some of those clones become memory B cells, right?
Yes, they do.
That's how we get long-term immunity.
That's right.
Like when you get vaccinated.
Exactly. That's the principle behind vaccination.
So these memory B cells, they don't immediately start making antibodies.
No, they do.
But they remember the antigen.
They do, yeah.
So if the body encounters that same pathogen again, those memory B cells can jump into action.
They can, yeah.
Much faster than before.
And mount a much stronger response.
Okay, so the second time around, the immune system is ready.
It is, yeah.
That's really cool. It's a very effective system.
Okay, so let's talk about the antibodies themselves for a minute.
Okay.
The guide describes their structure.
Yeah.
2 heavy chains, 2 light chains. Right. With a specific antigen binding site and a constant region.
It's like a Y shape.
Right, a little Y.
Yeah.
So the variable region at the tips of the Y that forms the antigen binding site.
Exactly.
And that's unique to each antibody.
Yes, it is.
It's like lock and key.
It is, yeah.
And then the constant region, the stem of the Y,
Right.
that determines the antibody's class.
Yeah, yeah.
and its specific function.
Exactly.
Okay, so what are the main functions of antibodies?
Well, the guide lists 5.
Okay.
Neutralization, agglutination or precipitation, complement activation, and opsonization.
Right, let's go through each one.
Okay.
Neutralization.
Okay.
That's basically blocking those pathogens from infecting cells, right?
Exactly. Antibodies can bind to viruses or bacteria and prevent them from attaching to host cells. So they can't get in and cause an infection.
So they're neutralized.
Right.
Agglutination and precipitation.
Yeah.
Those are the ones where the pathogens or antigens are clumped together,
Exactly.
making them easier for the immune system to clear.
Agglutination happens when antibodies bind to multiple pathogens at the same time, forming these big clumps that are easier for phagocytes to engulf.
Like they're all stuck together.
Yeah, exactly.
And then precipitation is similar.
Right. It is, yeah.
But it involves those soluble antigens.
They come out of solution and form these larger complexes.
So it's easier for the body to filter them out or for phagocytes to engulf them.
Exactly.
Okay, and then there's complement activation.
Right.
Antibodies can trigger that whole cascade
Yes, they can.
of complement proteins that we talked about earlier.
When antibodies bind to their targets on the surface of a microbe,
Yeah.
it can start that classical pathway of the complement system,
Right.
leading to lysis of the microbe, enhanced inflammation.
And increased phagocytosis.
So the adaptive and innate systems are working together here.
They are, yeah.
And then finally, there's opsonization again, where antibodies coat pathogens, making them more visible to the phagocytes.
Exactly, like putting a big target on their back.
So it makes them more attractive to those phagocytes.
It does, yeah.
Okay, so those are the main functions of antibodies.
Right.
The guide also goes into the 5 main types of antibodies.
Yes.
IgG, IgA, IgM, IgD, and IgE.
Each with their own special roles and locations in the body.
Yeah.
They're a fascinating bunch.
So IgG is the most common one.
It is, yeah.
And it can actually cross the placenta.
That's right.
Giving newborns that passive immunity.
Exactly. It provides protection to those newborns in his first few months of life.
Right, before their own immune system is fully developed.
Exactly.
Like a little gift from mom.
It is, yeah.
And IgG also plays a big role in activating the complement system, right?
Yes, it does. Okay, so what about IgA?
Well, IgA is found in our secretions,
Like
tears, saliva, mucus.
Right, so it's that frontline defense.
It is, yeah. It's protecting those areas that are most exposed to the outside world.
And the guide mentions that its levels can actually be reduced by stress.
They can, yeah.
So that's something to think about.
It is.
Maybe that's why we get sick more easily when we're stressed out.
It could be, yeah.
Okay, what about IgM?
IgM is usually the first antibody that's produced during an infection.
Okay, so it's the quick response team.
It is.
It's really good at agglutination and activating the complement system.
It is, yeah.
So if you find IgM, it usually means there's a recent or ongoing infection.
That's right.
Okay, and IgD?
IgD is mainly found on the surface.
Yeah.